your exam that is taking place on Monday. All right, we're going to start. So 1.2 is your terminology, and I have emphasized this like a million times. Please try and make sure that your spelling is correct. There is a marking rule that we apply if your spelling is not correct, but it doesn't mean if it's not correct, we'll give you a mark automatically. It depends. So when we say out the word, it still needs to sound like the word that we are looking for, and it should not sound different or mean something else in life sciences. So when you are studying, you need to physically practice writing the correct spelling for this section specifically. Okay, so here there is 8 statements. You just need to write the biological term. So here it says a section of DNA, a section of a DNA molecule that codes for a specific characteristic. That is a gene. So that is simple. And then a pattern of black bars resulting from DNA analysis. That is a DNA profile. And then evolution characterized by long periods of no change, alternating with short periods of rapid change. That is called punctuated equilibrium. So I need those 2 words for you to get a mark. And then the production of a genetically identical copy of an organism using biotechnology. This is under genetics. That is cloning, where we make an exact copy of a particular organism. We manipulate the genes and then make the exact copy. And then undifferentiated cells in animals that have the potential to become any type of tissue. That is stem cells. And the mark is at cells, stem cells. A genetic disorder that results in a person who cannot distinguish between the colors red and green. This one is easy. Color blindness. A part of the plant where the male gametes are produced. A part of the plant where male gametes are produced. That is an anther or a pollen sac. Actually, this is not really covered in the grade 12 syllabus, but I guess there it is mentioned. It's more of grade 11. So remember, we speak about meiosis taking place in animals and meiosis taking place in plants. So in human beings, meiosis will take place in the testes of males and take place in the ovaries of females. Then in plants, meiosis will take place in the anther for a male. So let's do it like this. So in a plant, meiosis in a male will take place in a part of a plant called an anther. Then in females, it will take place in the part of the plant called an ovary. So the part is the same for plants and also animals. But for the male, the part is different. In humans, it is the testes, and then in plants, it's the anther. Okay, I don't even remember mentioning it in the lessons. We actually never emphasize it at all. Okay, then the structure that joins 2 chromatids together, that is the centromere. And remember, there's these words now that confuse us in the exam because they sound the same and they look the same. So centromere, centrosome, and centrioli. So they mustn't confuse us. I think there is another one. So centromere is the one that joins the chromatids, and then centrosome is an organelle that produces the centrioli. Centrosome. I think I'm correct. I don't know why I'm doubting that statement. I am doubting it, but I think that is correct. Okay. And then, yeah, any questions on this one before I move?
Ma'am,
Yes.
would we need to study that part in 1.2.7?
It's not really part of our syllabus, but I would say it's just one word to study. Yeah. I never come across it, so I would say it's just one way to study, I guess. That's it. Where meiosis takes place, in a plant, in a male part of the plant, to produce male gametes. That's the anther. So this is the word. That's it.
Okay, ma'am, thank you.
Thank you. Anyone else, guys? Please also use the chat. Just click chat and then send a message. Then I will gladly respond to it. Yes, I'm listening.
Um, on question number 3, will I get a mark if I said punctual equilibrium?
No. Because remember, if your word is wrong, let's say the spelling or something, right? We need to pronounce the word. The word we are looking for, punctuated. But now if I read your word, it sounds what did you say? Punctual.
Yes, yes.
So that's two different things. Punctuated, punctual. So it sounds like two different things. So no, we're not going to give you a mark for that.
Okay, ma'am, can I ask something?
Yes.
1.2. If I said DNA profiling,
A pattern of black bars resulting from DNA analysis. I'm trying to think what the argument was on this here. A pattern, because I think I could be wrong. I need to remember it properly. DNA profiling is the process. That's how they explained it. It is the process whereby we extract these black bars. The black bars that we get at the end is a DNA profile. The process of extracting them from a sample is DNA profiling. That's how they explained it last time. I'm trying to remember if they ended up giving a mark or not. Yeah, my mind is failing me. I think this paper was 2020, what, 4? So I would say let's stick to DNA profile. How I understand it, the profiling is the process of extracting. Then you're extracting the DNA and getting the black bars. The black bars themselves, that printout that we see, is a DNA profile. So I think I will stick to that. I didn't see who was asking. Okay. All right. Anyone else, guys? Oh, okay. All right, let's move. Remember here, the goal is to try and confuse you. And if you do not know the content here, yeah, you are going to mess up. Remember, I've been strict on this and I said, here, the answer is worth 2 marks. You are given 2 options for a particular statement. And then from those 2 options, it is possible that only one option is correct. That can be A only or B only, or both options are correct, both A and B, or none of the options that you're given are correct. So you need to read the statement and understand exactly what they are speaking about. And you don't just look at 2 words and then try and answer. Read it twice. I would say 3 times or twice. The type of dominance in which neither of the 2 alleles is dominant over each other, resulting in an offspring with an intermediate phenotype. Is it complete dominance? No. Is it codominant? No. So the answer here is none, because the correct answer is incomplete dominance, and we do not have incomplete dominance from the options that are given. So the answer is none. All right. Then 1.3.2, the point where 2 chromatids overlap during crossing over, it should be A only. I'm just double checking. Yes, A only. So that's the chiasma, when 2 adjacent chromatids cross over during the process crossing over. And then locus is a position of a gene in a chromosome. So B is 100% incorrect. So our answer is A only there. Then a scientist who proposed the law of segregation, is it Darwin or Lamarck? It's none. The scientist that proposed the law of segregation is Gregor Mendel. Can you see how you could have easily chosen Darwin or easily chosen Lamarck there? Because you know, these are also scientists that we've studied about. But Darwin is the natural selection scientist, and then Lamarck is the law of use and disuse, giraffe, long neck, whatever business. So the answer there is none. Each answer is worth 2 marks. Can you see that if you've chosen a wrong answer here, you automatically lose 2 marks? So I need you to read the question twice and then understand exactly what they are looking for. I'm happy with this. Any question? I can take one question. Okay, we move. All right. This one, I'm gonna, yes, I'm listening.
Ma'am, why are they not putting none there? Because they are going to confuse people. Or is Vela is the plan? Like they've written complete 1.3.1. They've written complete dominion and co-dominions. Why can't they say C, maybe none?
Okay, so let's come here on top. Let's come here on top. So here, the instruction, I'm reading the instruction. Indicate whether each of the descriptions in column 1, the 2 descriptions that you are given A, what am I saying? In column 1, this is the description in column 1, applies to. You need to indicate if this description that you are given in column 1 applied to A only. What do we have on A only? Complete dominance. Or B only, that's what it says, right? A only there, or B only, meaning the statement only matches B, which is codominance, or both A and B, meaning the statement matches both the options, meaning complete and complete and codominance, or none. So none is given as one of the options there. So if you are choosing none, you are saying B and A are not matching the statements on column 1. So that is why it's an option. That's why they don't put it here, because they have not even put both A and B as an option here. So you just need to be able to look at the statement that you are given. Is A applicable to the statement? B applicable? Both A and B are applicable, or none, meaning A and B, both of them are not applicable. So we are following the instruction that we are given. They want us to answer it like that. Okay. And it is possible in an exam, like you can see, for both options to be incorrect. But do not jump to answering none, because 4 options, and do not skip this question, or do not leave any empty space, because you are given 4 options. You either choose A only, B only, both A and B, or none. So whatever answer that you choose, you have a 25% chance of getting it right. But let us choose the correct answer so that we can get that 2 marks. Okay, I hope that helps. The easy peasy questions I will ask you guys to do. Okay, but I think we've done this one. Let me do this one quickly. Okay, so this is from our first topic, DNA, obviously. So the diagram below shows 2 types of nucleic acids. Now this takes us back. When we speak about nucleic acids, we are speaking about DNA and RNA. There's only 2 nucleic acids. And we say these are very big, I would say, molecules. They call them molecules in life sciences. And they are made up of monomers. I think we've covered that. Then 1.4.1, identify the nucleic acid shown in diagram 1. So between DNA and RNA, the nucleic acid shown will be DNA. You are allowed to write DNA, or if you want to be fancy, you can write the full word deoxyribonucleic acid. But with exam stress and exam anxiety, you might end up messing up that spelling. So if you want to try to write the full name, but your spelling becomes incorrect, we are going to penalize you. So there's absolutely no need for you to complicate your life. Just write DNA. And your letters D and A should, all 3 of them, be in capital letters. So we accept this, but we're not going to accept that. I hope you are taking note. Same with RNA. So I need capital letters there and not small letters. And then Question 1.4.2 says, identify A and B. A here is showing us the bonds. Those lines are showing us the bonds between the bases because this will be a base and this will be another base. So A is your hydrogen bonds. Hydrogen bond. In most nodes, you will still get weak hydrogen bonds. That's correct. I think Swelihle also sent me a message about this today. So whether you say weak hydrogen bonds or hydrogen bonds, it doesn't matter whether you include the word weak or not. We're not going to penalize you. But the word that we are looking for is hydrogen bond. Okay, those are bonds that we find between the bases for DNA, your A, T, C, G. Then B, what is B? So if this is our base, this will be a sugar, that will be a phosphate. So B is your phosphate. Remember, these are our nucleotides. A nucleotide, one nucleotide is made up of a phosphate there in a circle, and then a sugar, and then a base. So same applies here. This would be one nucleotide. That would be a phosphate, that would be a type of a sugar, and then this would be a base. That can be either A or T or C or G, since it's in a DNA. Okay, and then name the type of sugar found in the nucleic acid represented in diagram 2. The nucleic acid represented in diagram 2 is your RNA, because we can see a single strand. Diagram 1 is a DNA. We've already answered that in 1.4.1, and we said that that is DNA because it's double-stranded and there's bonds in between, right? So the type of sugar that we are finding in diagram 2, firstly, you need to identify what is diagram 2, that is RNA. So meaning the sugar that we find there is ribose sugar. The tick will be there at ribose. Then which diagram, 1 or 2, represents a nucleic acid? So we've already identified that diagram 1 is DNA, diagram 2 is RNA, right? Then the question is saying, identify which diagram, 1 or 2, represents a nucleic acid that is used for tracing of human ancestry. So when we trace our ancestors, what do we basically use? We use the DNA. So that will be diagram 1. So to know a lineage between maybe a father, grandfather, great-grandfather, we are going to use a DNA, right? So that will be diagram 1. And then for paternity testing, so when we're doing paternity testing to try and find out if this particular male is the father, do we use a DNA or do we use an RNA? We use a DNA. So what diagram is it? Diagram 1. 6 marks. Okay, questions. I need questions, guys. I need questions. I think time to be shy now is over. Like really, it's over. I'm listening.
Ma'am, does it matter if you say phosphate group?
Phosphate group, that's fine. So we'll give you a tick there by phosphate. Phosphate group is correct.
Okay.
We'll give you a tick there by phosphate.
Okay, ma'am. Thank you.
Thank you. And yes, I'm listening.
I didn't understand why you are writing diagram 1 there at the question that says tracing of human ancestry.
Okay, so when we speak about tracing of human ancestry, we are looking at tracing down our ancestors. So, and in this case, we are not speaking about, well, directly, indirectly. We're not speaking about, I need to worship my ancestors, I need to slaughter a cow kind of vibe. Not that, right? We are speaking about a lineage, more like a family tree. So if we were to trace my ancestors, we would need to use my DNA to find out information about maybe let's say the parents, and then we can take it down to the grandparents and then take it down to great grandparents or whatever. So in finding out that lineage, we use the DNA, we use the DNA of human beings to find that. So the reason I said diagram 1 is because diagram 1 is representing the DNA. That's what we use to trace a particular lineage for a family. That's what I meant.
Okay, thank you.
Thank you. One more question, guys. Okay, I'm moving. Woo! This, I don't want to lie to you. Okay, I need your answers in this one. I'll give you 4 minutes. I told you I'm going to pressure you a little bit when it comes to time. I need 4 minutes. I expect you to be able to get 1.5.1 to 1.5.3. I expect you to get full marks there. That's the expectation. If you get 1.5.4 wrong, that's fine. That was considered a difficult question at the marking center. So I'll shush. I'll give you 4 minutes. But try your best to answer 1.5.4. Let me show the whole thing. If you can't see the whole screen, just please open the question paper on WhatsApp. Okay, I need answers. I need you guys to give me answers for this one. Okay, please raise your hand so that I can just call out people's names to make this process quickly. Let's do this quickly, guys. Please raise a hand so that at least I can select people's names instead of me calling out names and then people don't answer. Okay, Kamu, please give me your 1.5.1 answer. Name the type of mutation that caused or that occurred during the production of this gamete.
Ma'am, I got non-disjunction.
Non-disjunction. Okay, that is correct. We can be specific, non-disjunction. Anyone else with a specific type of mutation here. Non-disjunction. Non-disjunction is correct. The specific type of mutation here is chromosomal, because remember we've got two types of mutation. Mutation that involves genes, that is a gene mutation, and a mutation that involves chromosomes, chromosomal mutation. So the specific type of chromosomal mutation that took place here is non-disjunction. So non-disjunction they were giving, but the answer that they were looking for was chromosomal mutation, meaning this is a mutation that involves chromosomes. Yes, it was non-disjunction because chromosomes failed to separate. Okay. Let me see. I'm going back here. Okay, I need hands, guys. Hands. And we're not gonna move. If we don't do this quickly, we're not gonna finish the question paper. So because it's not about me yapping for 2 hours. Swelihle, what type of gamete is represented in the karyotype?
Sperm gametes.
Correct.
But I ended up saying female gametes because I noticed that there were 2 X's.
Okay, so you said sperm and then you crossed it out, and what did you say?
No, I initially first said female gametes.
Okay, when you say female, that's not a name of a gamete. That's a sex or a gender. When you say female gamete. Okay, so female, that's describing sex or gender. So if you were talking about a female gamete, that would be an ovum or an egg cell.
Oh, okay.
Okay, but explain to me why you said it's a female gamete egg cell.
Because there are 2 X's.
There are 2 X's, that is correct. Are they the same size?
No.
They're not. So if they are not the same size, then it's not a female gamete.
All
Okay.
right. Thank you.
Thank you. This was considered a difficult question. I'll read it here on top first. So remember, this is a karyotype, and the karyotypes that we normally do would be karyotypes with 46 chromosomes, showing all the chromosomes of a human individual. But here, this is a karyotype showing a gamete of an individual. So the expectation here is for us to have 23 chromosomes. But how many do we actually have in this karyotype? We have 24, because something happened here. Something happened. So the reason we are saying this would be a sperm is because, yes, you have gonosomes there, but they are not the same size. As much as it's X and X, they are not the same size, right? So if they were the same size, they would be a female. Since they are not the same size, that would be a male. So that is a sperm. That is why. This was considered a difficult question. And it's from a national paper. It's actually May. Okay? So if you're getting it wrong, but this one you should have gotten. You should have gotten the first 2. Okay, 1.5.3, A and B. I need someone to help us, please. Sinazo?
A, I said 23 autosomes.
In this gamete, give the number of autosomes.
22. 22.
22.
Oh.
And then gonosomes.
I said 2. I don't know because there were 2 X's there.
100%. 2. There are 2.
Okay.
Because where thereby 23, there are 2. In chromosome pair number 23, if we had all 46 chromosomes, we would have gonosomes, 2 of them. When meiosis takes place, we are expecting a gamete to have only 1 gonosome, meaning 22 autosomes plus a gonosome, or 22 autosomes plus a gonosome. That will be a Y. But what has happened here? Non-disjunction occurred. One gonosome, or that pair of gonosomes, failed to separate. So this is a gamete that is having 22 autosomes plus 2 gonosomes. This is an abnormality. Now you apply all your knowledge about non-disjunction. This is not a copy and paste from any karyotype that you've seen. You apply your information here. So there are two gonosomes. You cannot say one, because where do we find a gonosome in a gamete? We find it on chromosome number 23. I hope that makes sense. This was a little bit challenging. I need two people to give me answers for 1.5.4. I need to hear what you guys said before I answer. And remember what I said, this is already a challenging question. So do not overthink your answer. Just say what you've answered and then we take it from there. Anyone? I don't see hands. Anyone, please? Okay, I'm gonna call out people's names. And remember, we're not moving up until I get answers, eh? I need two people to answer for me. Renee, I'm listening.
I just said that the sex is female. That's all I wrote because I wasn't sure.
Sex is...
Oh, sorry, I said it's male, ma'am. Sorry.
You said sex is male. Okay, that's one answer. Okay, thank you. Let me take someone else's answer. I'm listening, Mapule.
Hi, ma'am. Okay. I
Yes.
also said it's a female, and then using X and Y representation, write down the sex chromosomes of the offspring. And then I did some bit of a calculation. I don't know if that's okay. So
What did
that
you get on your calculation?
the two X's and Y.
So how did you write your two X's? Did you say X, X, Y,
X,
or
X,
two?
Y.
Okay, you said X, X, Y, and?
Yeah, and then I just left it there.
You just left it there, okay? All right. I need two more people. Can I get two more people for this one? Cleo, I'm listening.
Ma'am, I think it will depend on the maybe who's stronger than the other one. Is it the female or the male?
Stronger in terms of what?
Like genes.
Okay, so what's your answer? Did you just say it will depend?
It can be a male or it can be a female.
Is that your answer?
Yes, ma'am.
Is that what you wrote down? It can be a male or female. Okay. I need one last one. I need option D. I'm going back to teams. Anyone? Kolile?
I said it's XY sperm plus X egg is XXY.
XY sperm plus, what did you say, plus X?
X egg
Yes.
then is equal to XY.
XY.
Now
Okay.
XXY.
XXY. Okay.
Mm.
Okay. Thank you, Kolile. What we're seeing here, option A is saying sex is male. I think I canceled this. Sex is male. B says XXY. C says it can be a male or female. D, it says there's an XY sperm and an X egg, and we get XXY. All right, let's work it out together. The gamete, this gamete fuses with a normal gamete. This information is important. This particular gamete that is shown in our diagram fuses with a normal gamete, meaning this gamete is abnormal. It is going to fuse with a normal gamete. Using X and Y representation, write the sex chromosomes of the offspring. Now, what is it telling us here? So what we have here is an abnormal gamete that is already, I'll write it here, abnormal gamete. We've already identified the gonosomes. We said the gonosomes that we have in the diagram is X and Y. So what do we have there? That is the abnormal gamete. It's X and Y. Then it fuses with a normal gamete. Okay, let me break it down here again. So where are we getting this X and Y? This is a gamete that is having 22 autosomes with 2 gonosomes, which is X and Y, because we're saying X and Y because they are different sizes, right? Plus a gamete that is normal. Since this gamete is already representing a male, but it is abnormal, right? And I know my kids, they were arguing, but ma'am, if it has both, why are we saying male and not female? Because it has both. That's too technical. We just take it as a sperm for now. I don't want to complicate anyone's life. Then a normal gamete, if this is a male gamete, it's going to obviously fuse with a female gamete. A female gamete will have 22 autosomes with 1 X chromosome. Now, in this side, I have not indicated the numbers, but this side I'm indicating the numbers so that at least it makes sense. This is our abnormal gamete. It is fusing with a normal gamete. So what are we expecting at the end? It will be these 22 autosomes plus these 22 autosomes, which is 44, right? And then we are also adding, it will be this XY, it will be added with this X gonosome. So what do we end up having here? X, Y, X, or 44 autosomes with X, XY. It doesn't matter how you arrange these ones, but they did not want the number of autosomes. So what are we going to then mention here? They said we must use a representation of X and Y to answer. So this particular answer is correct. This particular answer is correct. So what I have done here is to just explain the background to say, what do we normally have in normal fertilization? An egg cell will have 23 chromosomes, a sperm cell will have 23 chromosomes, right? And then we end up having 46 chromosomes because we would have added the numbers. So this is the same as a sperm cell can be 22. Okay, I'm not sure what's happening with my pen. 22Y for this particular one, plus with an egg cell 22X. When we add the two, we get 44XY. This will be a male because remember, we are just adding the chromosomes. In this case, we had an abnormality and they are asking you to then fertilize this normal gamete with this abnormal one. You are taking what you have. What do you have already? You have an XY gamete. Then you're going to add it with a normal one. The reason we are adding it with an X, a female gamete only brings in an X chromosome. It doesn't have a Y chromosome. That is why we are adding it with an X. When you add these together, you either get X, XY, or you could have written it as XYX, 2 marks for just doing that. This was considered a level 4 question. So level 4 meaning there's a very, very low percentage of learners that would get this. So if you did not get it, please don't feel bad. In a paper, there's what we call cognitive levels that will balance it out, right? So there'll be your high level questions and then your mid and then your low level. So low level questions, that's your 1 mark questions. We expect 99% of the learners to get it. So please, that is why we balance out these things. I don't want you to feel bad because now your answer is incorrect. So this was also challenging, to be honest, for you. Even for other teachers, they did not even understand what is happening here. So I'm happy that we're doing these difficult questions so that we can address this thing. Okay, I'm happy to take questions on this. I'm listening. Yes, I'm listening.
Ma'am, is it okay if you can please go back to the graph?
Yes.
So ma'am, if the other X is shorter than the other one, it means that it's a male.
Definitely. If it was female, if it was representing XX, we would have these two replicated chromosomes being the same size. So this would be an XX. That would be representing a female.
Okay, ma'am.
Yeah, I know. There was an argument about this. But yeah, because
But
they are the
then,
same size. Yes, I'm listening.
sorry, ma'am, but then you get a situation where it kind of looks like a string. I don't know how to explain it, but I've come across a few questions.
Where it looks like a strand like this. Like
Yes.
that? Okay, so remember the difference between these chromosomes and these ones. These ones are unreplicated. So remember, this is an unreplicated chromosome. It has not undergone DNA replication. So that's why it's easy for you to identify. So if it were to undergo DNA replication, this is what would happen. So this first chromosome has longer strands and this second chromosome has shorter strands. So what I've drawn here is actually very similar, if not the same as what is done there. Because those are still two chromosomes, but they are not the same size.
Okay, ma'am, I understand. Thank you.
Yeah, thank you. Okay, can we move, guys? I think we can. All right. Ooh, there is your evolution. I'll do this one with you guys, and then we move to another question. So I'll do this one. What do we have here? So if you feel like my screen is maybe projecting the question paper way too small, just view the question paper on WhatsApp. So we are doing 1.6. The diagram below shows the evolutionary relationship between different species. I'm trying to zoom in. Okay, there you go. Between different species. So this is an evolutionary relationship. So that's a phylogenetic tree or a cladogram between different species. So we have a common ancestor, and then we're seeing something, yeah, there's names of species and blah, blah, blah. Identify the type of diagram. The type of diagram that is shown above is what we call a phylogenetic tree. We addressed it in the evolution lessons. If you do not want to say phylogenetic tree, you can say cladogram. It means the same thing. Cladogram. It means the same thing. Okay, so that's a phylogenetic tree. And please don't say don't say phylogenic, genetic, not genic. All right. And then 1.6.2, give the genus that gave rise to the Australopithecus at A. The genus that gave rise to the Australopithecus at A. So A is the first genus after which one? There, that's the oldest one. So if you are tracing it from a common ancestor, this is the first one that originated, and then there was a branch. We first then had A, Australopithecus, and then B, we had our Homo genus. And then here still, it's still our Homo genus because that's Homo sapiens, blah, blah, blah. So these ones with an age would form under one genus. Remember, we did genuses. The other one that's not given here is that Adipithecus one. So how you read it, that would be from the common ancestor, and then that would be the gorilla. I'm wrong. It can't be this one. So common ancestor, gorilla, sorry. So the gorilla, sorry, let's rectify it. So this is where it starts, and then you start here. And then this would be a branch between the two. So the first one, gorilla belongs under Adipithecus. So yes, the answer is Adipithecus. I'll probably mess up the spelling. I need to check. I need to check the spelling there. Adipithecus. I rectified it, guys, please. So we move from the common ancestor, and then this is the first line of descent. And then you go up, and then here there was a split. So there was a split to get Australopithecus and Homo. And then this P, I think it's para something. I don't even remember it properly because it's not really covered in the syllabus. So I've rectified it. The reason you cannot write G gorilla, because you need to remember the genus. The question is asking us the genus. Another thing, I've mentioned this in the evolution lesson. This is a scientific name. When you are asked to write a scientific name, which consists of a genus name and a species name, you write the name and then you underline it. So this Adipithecus word, if you write it like this, Adipithecus in an exam, and not underline it wrong, you will not get a mark. A scientific name you need to underline. That is why on this statement, this is a scientific name. Australopithecus is a scientific name, but it looks different from the other words because they've done what? They've put it in italics. So scientific names, when you type them down, they will be in italics. When you are writing them down, you need to underline. Please take note of that. Please, please. Okay. And then give the letter that represents the most recent common ancestor for both, most recent common ancestor for both Homo sapiens and Homo erectus. That will definitely be, so where are we originating it? So here, then there, that would be the branch, and then we're at C. At C, we had a branch for erecta, we had Neanderthalensis and sapiens. So the letter would be at C. So that point at C, we then were able to get 3 branches, the branch for erectus, for Neanderthalensis, and then for Homo sapiens. This is not something you do once and you understand. I also need to double check my answers actually with the memo, because it needs a little bit of practice for you to get it. So the letter is there at C, because that is where Homo erectus originated, Homo neanderthalensis, all of them, they are from this branch at C. Okay. You would need to do a lot of practice for you to get this. Okay, that's why I'm also having my answer in my mind, and then I have to double check with the memo. Then 1.6.3, name 2 species that existed at the same time as Homo erectus. Where is Homo erectus? I need to see my lines, so I'm going to erase. So here, you would need to use your evolution content if you still remember the years, but you can also use your phylogenetic tree. So what we are able to see here is also the lines are representing how long, even though we don't have the timeline here million years ago, of the organisms, you'd need to use your lines so that you can figure out. So as much as we had a branch here, this means that the gorillas had lasted for a very long time. So if you answer the gorillas, you get a mark because that would still be in the same line. Let's see how Homo erectus there. Okay, so we still get gorillas, and then we still get that P troglodytes. Yeah, we still get it. If you answered Homo neanderthalensis, they also give you a mark there. I don't think they gave a mark for Homo sapiens. So they would say, okay, if you look at this line over here still branches the same way or at the same position as Homo neanderthalensis. Please make a note. They've said here, name 2 species. You are looking at the diagram. So what will be your answer? That G gorilla, that is a scientific name. So you write G dot gorilla, you underline. You are writing P troglodytes, you underline. Homo neanderthalensis, that's H neanderthalensis, you underline, because these are scientific names. So if you're not sure whether they're scientific names or not, they are in italics. Even in that phylogenetic tree, you can see they are in italics. So please take note. Other years they penalize, other years they don't penalize for underlining, but please just be safe. Okay, I'll take questions now. If you have a question, let me do 164 and then I'll take your question. Give one example. This question is so famous. Give one example of the fossil of Australopithecus africanus that was found in South Africa. There's 3 of them. It's Dawung Child, it's Mrs. Place, it is Littlefoot. Dawung Child, Mrs. Place, Littlefoot. Just double check. Yeah. All right. I'm happy to take questions. I'm listening. I think there was someone speaking. Mapule?
Yes. I want to ask something on that question number 1.6.3. So if I wrote only like G gorilla and then I don't underline it, will I be penalized for it?
Some years they have penalized, yes.
Okay. So we must remember to underline.
Remember to underline. I can tell you last year they didn't penalize, but it depends who's the boss that year. So just remember to underline. That's it.
Okay.
Ma'am,
Okay.
in 1.6.2, they didn't underline the Australopithecus. Why they didn't underline?
Oh,
Then they
okay. I've
underline.
said that a scientific name, when it's typed, it will be in italics, it won't be underlined. So if you look at the scientific names there, it's in italics, 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 all of them. When we answer, we underline because we are not typing, Anish. We are not typing. So when we answer, we write our answer down, we underline. But in the question paper, they will not be underlined, they'll be in italics. That is why.
Okay, ma'am. Thank you.
Thank you. These are things that make students lose marks, guys, because if a teacher is not mentioning these things, a learner will get out of the exam room thinking everything went well, and then at the marking center they get penalized for these small things. Like I'm saying, some years they penalize for not underlining, some years they do not penalize. Last year they didn't penalize for November and May. I'm not marking this year in May because I won't be around, so I do not know, but my colleagues will tell me. All right, this one, I think in every evolution question paper that we've come across, it has been asked. I am not saying it will be asked on Monday. I do not know. I do not see the paper or whatever. So let's just study. The phylogenetic diagrams, they are not easy, guys, especially if there's no timeline. Like I also have to double check. I could get it wrong. I got the first one wrong anyways. Okay, this we have done. This is our protein synthesis, but you guys easily forget protein synthesis. So it's your turn to answer. I'll be nice. 7 minutes. 7 minutes to answer this one. Okay, we're gonna mark. I'm not sure if the 7 minutes has elapsed. Can we mark, guys, or do you need 1 more minute? We can mark. Okay. Let's do this. We've done this before, but let's do it. This is under DNA, the topic for DNA. This is specifically protein synthesis. So I need to get my pen. The diagram below represents a stage during the synthesis of a certain protein. So they give you a diagram here. We can see the names of amino acids. And then here, that is obviously transfer RNA. And in a transfer RNA, remember, we get our anticodons. So these would be anticodons. Okay. And then here we have a strand. And here we have an organelle, right? This is obvious that it's a strand of messenger RNA. That is obvious. Remember our normal sequence, guys, when we're doing this. We first use a DNA template in order for us to get messenger RNA codons, to form a messenger RNA strand. And then we are going to use that to get transfer RNA anticodons, which will collect certain or specific amino acids from the cytoplasm. A chain of amino acid forms a protein. This is the sequence of protein synthesis. So identify organelle 3, that is your ribosome. So I'll answer this one. So that is your ribosome. That's the organelle that is represented here. And then describe the role of molecule 1 during this stage of protein synthesis. Describe the role of molecule 1, and they are saying we must describe for 2 marks, right? Molecule 1, I've already identified, that is your transfer RNA. So transfer RNA, very important. It brings specific amino acids according to the messenger RNA or according to the codon to the ribosome. So it brings specific amino acid, it picks up specific amino acids in the cytoplasm. So this is the cytoplasm of the cell. And then this transfer RNA is going to pick, there's an amino acid in another one and another one and another one. There they are all around the cytoplasm. So it will go and pick up a specific one. But the specific one that it picks up at that particular time is determined by the messenger RNA codons. That's 2 marks. Pick up a specific amino acid from the cytoplasm, but you don't have to mention from the cytoplasm, but you can for understanding, right? That is determined or coded for by a messenger RNA codon. That's the 2 marks. How do they phrase it here? It brings the required or specific amino acids according to messenger RNA or codon. Where is it taking this amino acid to the ribosome so that it can form a chain on the ribosome to get a protein? I'll answer everything and then take questions. Give the sequence of nitrogenous bases at 2. At 2, what do we have there? Give the sequence of nitrogenous bases at 2. So there is 2, it's a transfer RNA. So we need anticodons here, right? So if it's 1 mark, that should be easy, 1 mark. So in a messenger RNA, we have G, G, A. So that means a base that is going to pair with G will be C. A base that is going to pair with G will be C. A base that is going to pair with A will be U. We cannot make a mistake of writing a T there. It can't be CCT because this is from an RNA molecule, messenger RNA, and we are forming another RNA molecule, transfer RNA. We cannot be making that mistake at this stage. So if you've made it now, it's the last time you're making it. We are not making it in the exam. Okay? We need to double check. If it was the DNA, we would say it's CCT, but it's not a DNA. Okay. Then give the sequence of nitrogenous base on the DNA molecule that coded for histidine. We need to get it. They want sequence of bases on what? On a DNA molecule that coded for histidine. Histidine is the amino acid. So we are moving from what we are given, which is amino acid, and we are moving back to get a DNA. So we are given the amino acid here, histidine. Where is it? There it is, right? There is the amino acid. And what are we given here? That is the anticodon. So where is the anticodon? It's here. So that is GUA. You cannot just say, okay, there is GUA here and then there are no bases. So in a messenger RNA, then you say G will be coding with C, U will be coding with A, and A will be coding with U. Your answer cannot be CAU because what have you answered here? You've answered a messenger RNA codon. That's not what they asked. They asked for a DNA nitrogenous base. So you are going to move now from messenger RNA. So if we take it back, GUA will be CAU for what? For messenger RNA. But what do we want? We want DNA. So we're going to pair those bases again. C will be paired with G. A will be paired with what? With T. And U will be paired with A. This is your answer, unless I'm wrong. I'm double checking. GTA, 2 marks. That's how you get it.
So,
You
ma'am,
need,
you.
yes,
Oh, sorry, ma'am.
yes, I'm
Oh,
listening.
so you first converted to CAU and then you converted again when they ask that kind of question.
Yes, we need to read and understand how they've asked it because what did they give us? I'm going to change my pen. So this is very important. They gave us an amino acid. That's what they're asking there. Give a sequence of nitrogenous bases on the DNA molecule that coded for histidine. So this is different to A because at A they said they want bases at 2. At 2, where is 2? This is a transfer RNA. So you are going to use bases of a messenger RNA. So in A, they've given us bases of messenger RNA, and we just need to find bases for transfer RNA. That's why we paired. We do not convert, we paired. We paired it once, and then we got our answer because we moved from here to here, right? So you have to write this down on your exam so that you can figure out. Then what have they done at B? At B, they're saying on the DNA molecule that coded for histidine. So what did they give us? They gave us the amino acid. So we are starting here on the amino acid and they want us to get a DNA. So we are moving. But now amino acid they've given us, transfer RNA they've given us, that is GUA. So we are working from actually GUA to get the bases of DNA. So we are going to pair these bases to form a messenger RNA strand first, CAU, and then use these bases to pair them to get the bases on a DNA molecule, GTA. So if your answer is CAU, that is wrong. You have given us bases of a messenger RNA and not the DNA. Okay.
Okay, ma'am. Thank you.
Thank you so much.
And
Yes,
sorry,
I'm listening.
ma'am. So are you meant to do it like that always when they ask a situation like that, unless they rephrase the question?
I'm 100% sure that they will not ask it like this again, right? They will trick you. You just need to know the sequence that we move from DNA to messenger RNA to transfer RNA, amino acid, and then a protein. So they can give you next time a messenger RNA and ask you about an amino acid. So it's a matter of understanding how you do this instead of just thinking if they ask me about this and they want that, this is how I do it. So this is the sequence. We move from DNA, messenger RNA, transfer RNA, amino acid, and a protein. It depends what they've asked you. They can give you this and ask you for that. So you need to move twice, depending on the diagram that you are given. Okay.
Okay, ma'am. Thank you.
Thank you. Anyone else, guys? Before I move to 2.1.4, anyone else that needs clarity? Okay. So 2.1.4 is a mutation question. during the synthesis of the same protein, meaning during the process of making the same protein that was made here in this diagram, there was a change. When we speak about a change, we speak about mutation. We did this in detail, right? And I said, this is your application question, because when we speak about mutation, we normally speak about gene mutation here in DNA, right? And then we speak about chromosomal mutation in meiosis. Here, if there is a change that has caused the basis of DNA to change, maybe the sequence will change in DNA. So if the sequence in DNA changes, that will impact the messenger RNA codons, that will impact the transfer RNA anticodons, that might impact the amino acid that has been formed. So if the amino acid formed remains the same, that means the same protein is going to be formed. That is important. If the same amino acid is formed, that means there'll be a same protein that is formed. If you don't understand this, you need to go back to the lessons. The links are still there on each group. But if an amino acid that is formed here is different, that means the protein that is going to be formed will also be different, because the protein formed is dependent on the sequence of amino acids. I did this lesson. I'm sure other people can remember. So what do we have here? During the synthesis of the same protein, there was a change in the amino acid sequence, right? Because alanine was then replaced by tryptophan. Explain the possible reason for this. So they're saying alanine, there is alanine, was replaced by tryptophan. So meaning instead of making a particular protein with the sequence alanine, histidine, tryptoleucili, right? This one then got replaced with just double checking. Yeah, tryptophan. So now what sequence do we have? Our initial sequence is supposed to be alanine, histidine, tryptoleucine. But what do we have now? Histidine, tryptoleucine. So what happened here? Mutation happened. 1 mark. Mutation occurred. Because what are they saying? They're saying we must explain the possible reason why now an amino acid alanine was replaced with tryptophan, meaning mutation occurred. But now, where did this mutation occur? Because once you say mutation occurred, that's 1 mark. And what is the allocation? 3 marks. So you are going to trace it back. If this confuses you, you need to go back to the lessons, right? So what do we have for alanine here? So in alanine, we had, I'm going to change my pen. Okay, so in alanine, when we were coding for alanine, we had GGG in transfer RNA, CCC in messenger RNA, meaning our DNA was GGG, right? Because we're going to move DNA, there is DNA. And then from DNA, we were able to form the codon CCC, and then we're able to form the anticodon GGG, and then which coded for alanine. But now, this alanine got replaced with tryptophan. We know the basis for tryptophan. So what are the bases for tryptophan? Anticodon is GAA, codon is CUU, and then that was the amino acid. So what happened here? Let's figure it out together. When we still had our alanine, we had GGG, which was our DNA, which coded for, I'm going to wait for my pen, which coded for messenger RNA CCC. There it is. Messenger RNA. I'm not sure if you can see what I'm writing, but I hope you can. And then this would have coded for an anticodon GGG there. That will be for transfer RNA, which then coded for which amino acid? Alanine. But a certain mutation happened here because we end up having tryptophan replacing. There is tryptophan. We are given the anticodon for tryptophan. So the anticodon for tryptophan is GAA. And the messenger RNA, I'm working backwards now. This can be a little bit confusing if you don't have the foundation. So I'm working backwards for this one. GAA, which has a messenger RNA of CUU. Let's see, GAA, CUU. Okay, so I'm still fine. That means the DNA here was GAA. So where is the mutation? So if you check the bases, the initial bases were GGG, right? But now that means mutation occurred whereby A and A were replaced. So G and G got replaced by A and A, which changed the messenger RNA. So the messenger RNA for that particular triplet was initially CCC, but because of a change there at the DNA, it ended up being what? CUU. And initially, the transfer RNA was GGG, which ended up being GAA. And all of this is because of a mutation that occurred in the DNA, which means that the initial amino acid that was coded for was alanine, but then because this anticodon codes for tryptophan, that's the amino acid that was brought forward. What does this mean for this particular protein? A different protein is going to be formed. So how do you answer it? You can answer it like that on the side, but you need to phrase it nicely for you to get the marks. The first mark, a mutation occurred, and everything that I've just mentioned, it's there on the memo. I'm going to share the memo with you guys. That a change in sequence of the nitrogenous base changed from what? From GGG to GAA. There's a tick for this. You speak about the nitrogenous bases, GGG to GAA. What did that mean for messenger RNA and for transfer RNA? There. And the amino acid coded for were different. And that meant that the protein that is going to be formed will be a different protein. This is not something you can just understand from this video that I'm just explaining now. You would need to go back to the lessons and do a little bit of practice also. Okay, let me take questions. Okay. This one is for you guys. Oh, I kind of skipped something. There's something that, okay, 2.2. I'm not going to ask you guys to write it down, right? Thereby 2.2. Remember, you need to be able to know how to describe DNA replication. You need to know how to describe transcription and translation. You need to know how to describe. You do not know which one they are going to ask you. And all three of them are normally about 5 or 6 marks, right? And in DNA replication and transcription, they are very similar processes. But the first 3 points, I think, are the same, first 2 or 3, and then the rest are different. Because remember, in transcription, we are using DNA to form a messenger RNA strand. And here, we use only one strand that is going to act as a template. So the DNA double helix unwinds, so the DNA double helix unwinds and the weak hydrogen bonds break and the molecule will unzip, right? And then one strand is going to be used as a template to form a messenger RNA. And then we use free RNA nucleotides from the nucleoplasm to pair to form that strand of messenger RNA. A will be complemented by U, C will be complemented by G. That is transcription, right? But with DNA replication, the first 2 points are the same. The DNA double helix molecule will unwind and the weak hydrogen bonds or the hydrogen bonds will break, forming 2 strands. The molecule will unzip, forming 2 strands. Both strands are going to be used as template. In DNA replication, we use both strands. In transcription, we only use one strand. Both strands are going to be used as templates to form complementary strands. How do we form those complementary strands? Free DNA nucleotides from the nucleoplasm will form those complementary strands. At the end, we will have 2 identical DNA molecules, not DNA strands. Unfortunately, you have to go back to that particular lesson. This one is for you guys. Yeah, we're running out of time. I think I'll just have to do these things, guys. We are running out of time. I don't want to be speaking till 8:30. So let me do it quickly with you. The diagram below represents metaphase 1 of meiosis. Now they've told us what phase that is. They're saying it is metaphase 1. They want us to identify part B. Part B is the circle over here. We know that will be the cell membrane. That can't be the nucleus. Because remember, by the time we get to metaphase 1, the nucleus would have disappeared in prophase 1. So that circle on the outside would be cell membrane. So if it was like this would be cell membrane, and then this would be the nuclear membrane, and then we have chromosomes. But we only have one cell, one circle there, so that would be the cell membrane. Then state one function of part A. Part A is the centriole. So the centriole forms spindle fibers. Yes, it's correct. It forms spindle fibers. These are the spindle fibers that it forms. Or you can say it produces spindle fibers. That is part A. Woo! Wow. 6 marks. Explain the contribution of I need to remember what the answer was here. Explain the contribution of metaphase 1 to natural selection. What have they done here? They have linked two topics. They are linking meiosis and they are linking evolution for natural selection. We first need to remember what happens in metaphase 1. And what we mean when we speak about natural selection, species that are able to survive, and species will obviously be different in the environment and all of that. So in metaphase 1, here we have chromosomes randomly arranging in the middle of the equator. And because of how they are randomly arranging in their homologous pairs, that is important, in their homologous pairs, that will also lead to the gametes that are going to be formed at the end. So how did they phrase this to answer the question? Random arrangement of chromosomes in the middle of the equator, that is the tick, results in the formation of genetically different gametes, tick. Now, if the gametes are different, what does that mean in terms of the population? If their gametes are different, that means the individuals that are produced in the population will also be different, because my DNA will be different to my sister's DNA, will be different to my brother's DNA, will be different to my cousin's DNA. That means there'll be variety of individuals in the population, and that is all caused by what? By metaphase 1, because the chromosomes arrange themselves randomly. The reason we are saying this results in different gametes, because initially they are arranged there in pairs, but in anaphase 1 they're going to be separated. This particular chromosome will form part of one gamete, and this chromosome will form part of another. Let me explain all the points. Random arrangement of chromosomes, tick, results in the formation of genetically different gametes, tick. This leads to an increased genetic variation in a population, tick, which will cause, since we are different as individuals, natural selection, because remember we speak about others surviving and others not surviving, right? Which will cause some individuals to have favorite characteristics and other individuals to have unfavorable characteristics. When the environment changes, what happens to those with favorable characteristics? They'll be able to survive and reproduce. What happens to those that have unfavorable characteristics? They will not be able to survive. They will die. Those are your points. This was not an easy, this was considered cognitive level 3, because now you are bringing in information about metaphase 1 and then bringing in information about natural selection. It was not easy. The diagram below shows 4 possible daughter cells formed at the end of meiosis. Remember, with meiosis, we form 4 daughter cells. And what are we having here? It's actually the daughter cells from the diagram that we have above, because the diagram that we have above has 8 chromosomes. And each cell here at the end has 4 chromosomes. Draw cell D, including the missing 4th chromosome. I would be very much interested to see your drawings. So what have they done here? You need to figure it out, okay? Because remember, what is going to happen in anaphase? These chromosomes are going to be separated, right? So we need to take it back, because these ones on this side on top are all going to move to the upper part of the pole. And then these ones are going to move to the bottom part of the pole to form the 2 cells at the end of meiosis 1. And then at the end of meiosis 2, what would have happened to these replicated chromosomes? They would have also separated. So that means at meiosis 2, this one would separate with this one. Because remember, at this point, they are replicated. In anaphase 2, they are going to be unreplicated chromosomes. So how are you going to trace this? Using this genetic material, the shading. Check the chromosomes there. I'm going to erase because this is important. So you need to check the shading there because this shading there is not there to just decorate chromosomes. This is actually showing us the genetic material that has been exchanged during prophase 1. So what are we going to do? In each cell, we will need to trace back a particular chromosome here. So in cell 1, we are having 3 shaded chromosomes. And then 1 unshaded. So what have they done here? Let's look at this. This is actually chromosome 1, chromosome 2. 1 is unshaded, 1 is shaded, fully shaded. I'm sure you can agree with me. So these chromosomes here on top are the chromosomes that have formed this cell. Because remember, we are first going to separate these chromosomes. I can't draw all of them at the same time. So this would form the first cell, and these ones will form the second cell. That would be telophase 1. And then they will undergo meiosis 2. So what will then happen? The replicated chromosomes are going to be separated to form unreplicated chromosomes. So the ones there on top, this one, this one, this one, this one. So 3 fully shaded ones and 1 unshaded, they form that cell. And then the ones here at the bottom, it's half shaded, half shaded, half shaded, and half shaded. These ones at the bottom have formed which cell? That one. I'm going to erase so that we can see which ones are shaded properly, which ones are not shaded. There you go. So the second line has formed which one? That cell. And then the third one, what are we able to see here? Half shaded, half shaded, half shaded, half shaded. These ones are forming the third cell. So what are we then given in the fourth cell? We should be taking these chromosomes here at the end. This one, with this one, with this one, with this one. So what do we have included here? Is this unshaded one there? We're expecting how many unshaded? Let's look. We're expecting 1, 2 fully unshaded, 3 fully unshaded. But what do we have in the diagram? Only 2. And there, where is the other one that we have as the fourth one? Fully shaded. There it is. So that means the missing chromosome is 1 unshaded chromosome. Oh, we had to work a little bit. So they're saying draw cell D, including the missing chromosome. This would be a drawing. So this is your cell membrane, and this would be your nuclear membrane, and this would be chromosome 1, chromosome 2. That's the chromosome that is unshaded, and then the last one that is shaded. You need to write a caption. Remember what I said about drawings in life science? Whatever drawing you write in life science, it should have a caption. So what do we then have here? That is, they've said the diagram below shows 4 possible daughter cells. So this would be a daughter cell. Tick for the caption. Missing chromosomes is unshaded. Cell D correctly copied. Okay. There was no marker located for the caption here, but I'm 100% sure they probably manipulated the memo. Your drawing in life science should have a caption. Okay, I'm listening, guys. We are running out of time. I'm listening. Any question for this? 233 was difficult. 234 was not. So that natural selection question was difficult. Okay. Okay, this one is for you. 5 minutes. This one is for you guys. Okay. Can we mark or do we need 2 more minutes? We can mark, guys. Okay. Let's mark. Let me just finish my order here. This is a pedigree diagram. I think we did quite a few pedigree diagrams. And also there's quite a few videos on the YouTube channel that you can check out. Remember here, to be able to answer a pedigree diagram, you will need a good foundation of the crosses, specifically the monohybrid crosses. I'm going to go back to my memo, specifically the monohybrid crosses. So you would need to understand these are not difficult at all. So I think this is the last question we're going to do. So cystic fibrosis. Remember how I encouraged you guys to carry a highlighter? So this is when you read the words and then try to get a bit of understanding of what is happening here. Cystic fibrosis is a genetic disorder caused by a recessive allele. This is very important. Remember in pedigree diagrams, I said there will be 3 types of pedigree diagrams, right? You can be given a pedigree diagram that is speaking about a certain gene or genetic disorder. Now, they will tell you if it's caused by a recessive allele, or if it is caused by a dominant allele. So that is the key information that you would need to know in this case, right? Because if it's caused by a dominant allele, so that means an individual with this particular disorder will need to have a dominant allele. But if it is caused by a recessive allele, that means in a heterozygous condition, it's going to be masked. So if this is the disorder, and let's say we use letter P, an individual, if the disorder is caused by a dominant allele, an individual will need to either be homozygous dominant or heterozygous dominant to have the condition, right? But if the condition is caused by a recessive allele, the only genotype for that particular person will be the small letters. I know with the P, it's difficult to tell if it's small or big. So the 2 small letters will be obviously the genotype for an individual with that genetic disorder. So you need to read into it. Or your pedigree diagram can be sex-linked. Remember, if it's just caused by a recessive allele or a dominant allele, we use normal letters. That would be a B or whatever letter that you are given. So the genotype will be maybe capital B, capital B, small letter B, or small letter B, small letter B, right? But if they say a particular disorder is a sex-linked disorder, we know that when we do our genotypes, that is when we use our XX and our XY, because it is sex-linked, it is linked to the sex. So we use the gonosomes there. And we are given a letter of that particular genetic disorder or sex-linked, they will say sex-linked disorder. So if it is hemophilia, it will have an age. If it is color blindness or whatever. I'm making a mistake here. One small mistake. The Y doesn't have the allele. We need to remember that. The Y will not have a superscript or an exponent. So that is very important. There are 3 types of pedigree. You need to identify which pedigree diagram you are given. If it's a sex-linked, they will mention it sex-linked, or they will give you genotypes. Once they give you the XXXY genotypes, that's when you know that is a sex-linked one. Very important. First question. And remember, we don't ignore the key, right? Because the key is very important. So what are they saying here? They're saying in this pedigree diagram, the ones with, I think this looks like a square that is not shaded. These are males without the genetic disorder, right? And the ones that are not shaded, circles, females without, and shaded square, males with shaded circles, male without. As much as we are still having the different genders in our key, this is not a sex-linked disorder. 2.4.1. How many generations are represented in the diagram? Generation 1, generation 2, generation 3. 3 generations. Anyone to question that? Anyone to maybe query why we are having 3? I think that is straightforward. Okay. Then give the genotype of individual 9. I need someone to give me the genotype of individual 9. Anyone? Please.
I said lowercase bb.
Lowercase bb. Anyone who said uppercase bb? Anyone who said uppercase bb? We are using letter B because they've given us letter B. Anyone? Sinazo, did you say capital letter B, capital letter B?
No, I said capital letter B and small case b
So
because
you
the
say
parent
because
because both the parents do not have that disorder.
so the parents being 5 and 6.
Yes.
Both the parents do not have.
Yeah, they could be I don't know because the disorder is recessive. So
Okay, wait. I need to understand your answer. So you are saying both parents do not have the disorder.
yes,
Okay. Is 9 having the disorder or not?
9 has the disorder.
9 has the disorder. Okay. So what do you think is your genotype for 5 and 6?
5 and 6.
Yeah. What do you think is your genotype there?
I think it's also okay. The thing says the disorder is a recessive.
Yes.
Oh, if it's a recessive, okay, now I'm confused because it's usually a dominant allele, and then if the child has it, which means the parents probably has a recessive allele of the disorder, meaning it's dominant over the recessive. So because now the disorder is recessive, it's tricky.
You love that. Love that interpretation. So where is this taking us back to? This is taking us back to this important information that I highlighted here. So if a condition or disorder is caused by a recessive allele, the individual with this disorder will need to have 2 capital letter B's because it is caused by what? By a recessive disorder. So this one has the condition. So this is an individual with cystic fibrosis. There's no such abbreviation. I'm just writing it there because I don't have space. Now, because they've said that this individual or disorder is caused by a recessive allele, meaning an individual with capital letter B, guys, this individual will be normal, will not have the condition. Because this individual is now having a dominant allele and a dominant allele. These would be normal alleles. What about an individual who is heterozygous? This condition is caused by a recessive disorder. But remember, a recessive disorder is masked or is dominated by a dominant one. So this would be a dominant normal allele. This would be a recessive cystic fibrosis allele. So this individual that is heterozygous is normal, also does not have the genetic disorder. They carry an allele of the genetic disorder, but they are normal. And the reason they are normal is because the normal gene is dominant over the cystic fibrosis gene. So that is the interpretation based on what we are given. Our most important information is the information that they've said that the genetic disorder is caused by a recessive allele. Then you write this down on the side. These ones are normal and this one is not normal. So if we go back to our question, the first answer that we got, which was small letter b, is correct. Why are we saying that? They're saying give the genotype of individual 9. Individual 9 is shaded. This is a female with the disorder. So any individual, male or female, with the disorder, what will be their genotype? Small letter B, small letter B. But what is this telling us about the genotypes of the parents? I need someone to give me the genotypes of the parents. I've explained it now in detail. So if individual 9 is having a small letter B and a small letter B, that means they would have inherited one small letter B from each parent. I need a genotype of 5 and 6. They actually have the same genotype. That's what I'll tell you. Anyone, please unmute and tell me. Genotype of 5 and 6.
Uppercase B and lowercase b.
100% correct. Because how do we know this information? We know this information because there's individuals here, these two offsprings from the parents 5 and 6, they have the genetic disorder. What does that mean? That means they have two recessive alleles, which is capital B, capital B. Now they don't just take, sorry, not capital, small letter B, small letter B. They don't just take these alleles from the atmosphere. They need to be accounted for, and each letter will be accounted for by each parent. This B from one parent and this B from another parent. So what do we know about this parent? These parents, they have a recessive allele, but based on the key, they are actually normal. So what is it telling us? They are heterozygous normal, because they cannot be homozygous normal, because if they're homozygous normal, they would have capital letter B, capital letter B. But now they are normal, they are carrying that allele that got passed on to certain offsprings. Very important. Very, 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 very important. Yeah, I hope that makes sense. Then 2.4.3. This is a little bit of practice also. It's not something that you'll get just after 2 minutes. It needs a little bit of practice. So please do me a favor. Whenever you are doing, now that you will probably be doing more pedigree diagrams, read your information and first identify for me if that is a genetic disorder caused by a recessive allele or dominant allele or a sex-linked pedigree diagram. Because this information will be different if we said that this genetic disorder was caused by a dominant allele, right? If we said that, I hope I'm not confusing anyone. If now I'm changing a color, if we say that this genetic disorder was caused by a dominant allele, so that means this one, capital letter B, will be an individual with the disorder because it's caused by a dominant. So dominant you have. So this individual will have the cystic fibrosis. And this one will also have cystic fibrosis because what? The cystic fibrosis allele will be dominant over the normal allele. It's the opposite now. An individual who does not have the condition will be the recessive one. So I hope you can see that it's important that we understand. I just don't have time, but I wish I could. I'm going to peruse through after the lesson and then send you a different pedigree diagram so that you can see this. So if the dominant allele was the one causing the genetic disorder, this would be an individual who is normal, who does not have the genetic disorder. And then these two would have the genetic disorder. Let's answer this. The next one. Explain why both individuals 5 and 6 are 4 marks. We've answered it. Explain why both individual 5 and 6 must be heterozygous for cystic fibrosis. Now, 4 marks. I need you to, if you have not answered this question, to just take 30 seconds to try and answer, so that you can see once I start mentioning the memo, you can tick your answers or mark them wrong, because we are not skipping the 4 marks. We are not skipping 6 marks. Why are you saying individual 5 and 6 are heterozygous? They've given you the answer. The reason they are heterozygous, both of them, is because the offsprings 8 and 9, their genotype is small letter b, small letter b. They would have inherited one small letter b from each parent, or they would have inherited the cystic fibrosis allele from each parent. And the parents are normal. If the parents are normal with this cystic fibrosis allele, that means one thing, they are heterozygous. They are carrying this allele, even though they are normal, they are carrying it. This allele is actually dominated or masked because of the presence of the dominant allele. The basics of genetics will need to come in handy in order to understand this. Let's read it from the memo. Both parents, 5 and 6, have a dominant allele and a recessive allele. That's why we're saying they are heterozygous. They have a dominant and a recessive allele. Since they do not have the cystic fibrosis, or you could have said normal here, they have children 8 and 9 with cystic fibrosis who received one recessive allele from each parent. This I need us to get. In pedigree diagrams, you will have 1-2 marks question, and then there will definitely be an explain question. You are explaining something. And what I need you to understand, any offspring, whatever letter they have, they would have inherited one from each parent. So if they give us a genotype of an individual who is like this is the genotype. What we know is one of the parents has a capital letter B, the other parent has a small letter b. We are not mentioning the gender to say the mother has, the father has, because in this case, we do not have enough information here, right? Even if an individual is having a small letter R and a small letter R, we know that each letter will be accounted for by the parents, even though we do not know the other letter, but we already know the ones that we are able to see. We will use other information in our question paper to know the other letter. That is very important. Okay, I'll take questions now. 2 hours in. Using the letters B, do a genetic cross to show the inheritance of cystic fibrosis when heterozygous parents are crossed. Using genetic letters BB, do a genetic cross to show the inheritance of cystic fibrosis when heterozygous, meaning 2 heterozygous parents. You do a cross. And how many marks? 6 marks. So I do not have space, but this would be your cross. You would have the phenotype, right? And in your phenotype, you will write, they're saying they're heterozygous, so they do not have. So it would be male without, because they said they do not have. If they are heterozygous, meaning they are normal, based on our information, yes. If they are heterozygous, they're normal. So you cannot say in your phenotype, heterozygous parent, no, that's wrong. So if there's parent, there'll be a male and a female, right? And since they're heterozygous, that means both of them do not have. So it would be a male without cystic fibrosis. And that will be crossed with a female. Even if you started with a female, it doesn't matter, right? The female, this female does not have also without cystic fibrosis. You write it in full. If you mention anything about heterozygous, no mark. And then you write your genotype. Even if we are still speaking about male and female here, it is not sex linked. So we're not going to use X or X and Y. So heterozygous, they crossed with this. And then you will do your, I forgot something, P1. And then you will do your meiosis, the capital B, small b, capital B, small b. And then this would be your gametes. You do a Punnett square for your gametes. I don't have space, that's why I'm writing it here. Capital B, small b, capital B, small b, capital, small, capital, this was what, a small one. Okay, small, and then small. My pen is lagging. I'm just double checking. Maybe. All right. Then your F1. F1, you write your genotype. Genotype, you have what? 1 is to capital letter B, capital letter B. 2 is to capital letter B, small letter b. 1 is to small letter b, small letter b. I'm doubting my things here. Okay, let me just double check. It should be right, but yeah, it's correct. Then your phenotype. Phenotype. This would be 3. This would be without. 3 without cystic. Where am I getting the 3? It's this one here, and then these 2. Both these genotypes represent individuals or offsprings without cystic fibrosis. And then you will say 1 with cystic fibrosis. Okay, ask me questions. Those that want to leave, if you want to leave, you can leave the meeting. The rest of the questions, we will answer them tomorrow. So please just continue answering from 2.5. Then we'll have to do a lesson tomorrow. I'll just have to try and figure out my day if I'll be back before 5, and then we can do a lesson. I'll confirm a time. Please ask a question. This was not an easy pedigree diagram. Please ask me a question. Okay.
Ma'am, when it comes to the parents of 5, would their genotype be uppercase B and lowercase b, or would it be between upper lowercase b and upper
For
lowercase
parents
b?
5. So
Yeah.
for parents 5, what we know is that one of the parents, so parent 5 would have inherited capital letter B from either of the parent and small letter b. So it is possible that one of the parents, so we cannot decide. That's why they didn't ask us any information, because they didn't give us enough. So what we do know though is that one parent has capital letter B and the other parent has small letter b. So what is this telling us is this parent can either be homozygous for normal or heterozygous. It is also possible, right? One parent. And then the other can either be heterozygous or recessive. There's 2 possibilities for each parent. But what we can account for is that one parent has capital letter B and the other parent has small letter b.
Okay, ma'am. Thank you.
Thank you. I'm listening, guys. Please, if there's anything unclear, please. Okay, we'll leave it at that. 2.5, up until the end. Please answer it. We'll do it tomorrow. I know this is tiring because you feel like you're doing the same thing. You've got notes to cover. You've got this to cover. I understand, but it is better that we do it this way so that at least you can, I would say, check your mistakes in a way. So please answer it. I know you do not have time, but that's the only way you're going to practice. So just try and answer 2.5 up until the last question. I think that is actually a big part of evolution. I will confirm the time. We can probably do 5 o'clock. If I end up not being back in the house, I'll send a message. But let's do 5 o'clock tomorrow. I've sent a link of the paper 2 topics outline, the same thing that I did on Thursday. So I'm not going to repeat that, where I go through everything and then remind you what you need to know for each topic. So you can watch those videos. They're available for you guys to catch up. Really, let's push. We can see that the papers, yes, as much as we know the format and we know what they're going to ask, but we sometimes not sure how they're going to ask it. Yes, others we know, but we're not 100% sure about everything. Investigations, I'm going to squeeze in 5 minutes for investigations because these I am passionate about. Let me get my pen. Okay, let me check if my order will be delivered now because the security will call me. Okay, I have 9 minutes. I have 8 minutes. When it comes to investigations, I spoke about this already, guys. I said that your aim is your friend. How do you get an aim? An aim is a statement that is telling you about the investigation. So if, for example, I'm going to scroll down, there should be an investigation. There'll definitely be an investigation. I just need to find it. Okay, there. They will always give you an aim. Once they say scientist wanted to, that is your aim. Highlight it and write aim next to it. Scientists wanted to determine whatever that they have after the word to determine relationship between, this will obviously be a very challenging one. But most of the time, they use the words effect. They wanted to determine the effect of this on that. Now, what do you have here? This will be a different one, and I will explain it tomorrow. They speak about the relationship between the presence of tall trees and the wings of stoneflies. The factor that is mentioned first will always be your independent variable. It doesn't matter how difficult the investigation is, because the first questions that you have here is identifying your variables. You cannot fail to identify your variables. So if, for example, my effect says, or my aim says, to determine, the scientist wanted to determine the effect of fertilizers on plant growth. I need someone to tell me the variables here. Give me your independent and dependent. I've taught you this, how to get those two variables from this statement. You don't get them anywhere else but from this statement. I need someone to give me an independent and a dependent variable.
Ma'am,
Yes.
the fertilizer is the it's a dependent variable, and then the plant growth is the independent.
Okay. I'm going to read and explain it again, and then I'm going to ask you, right? So last time I said what you want, the effect of what you want to investigate, that's your independent variable. So what is it that we want to
The
investigate here?
The effect
The
of
plant-
the fertilizer.
Not the effect. It is
It's
what?
the
The fertilizers. Remember I said,
Yes.
once you mention the word effect of, wrong in your answer. Have it in your mind. They want to investigate the effect of what? Fertilizer. After the word of, that is your independent variable. So if I am planting some things on my garden, and then I'm realizing that after I've added something happens to these plants. So what is my independent variable? Is the fertilizers that I'm adding, because I want to investigate the effect of them, the fertilizers. So that would be my independent variable. Now, the plant growth is what I can measure, because I can measure the plants growing, right? Now, whether my plants are growing or not is dependent on the fertilizers or on the effect of the fertilizers, even though we don't write the word effect. So the plant growth will be the dependent variable. It's always like this, right? Because if I add fertilizers on my plants, what do we expect? We are expecting them to grow, because now there's nutrients in these fertilizers, or to grow at a faster rate. But if I don't add fertilizers on my plants, they will still grow, yes, but they may grow at a slower rate. So whether the plants growing or not are dependent on what? On the fertilizers. I'm going to write another aim. To determine the effect of light intensity on the rate, I'm not sure if you guys are going to see this, on the rate of photosynthesis. I need my variables, my aim. 2, scientists wanted to determine the effect of light intensity on the rate of photosynthesis. I need variables. I need independent and dependent there. Anyone?
Independent light intensity.
Thank you. And your dependent?
It's a rate of photosynthesis.
Thank you. Dependent. Because the rate at which photosynthesis takes place is dependent on what? On the intensity of light. You give a plant high intensity of light, it's going to photosynthesize more. I hope you get it. Now, these are basic basics. This is grade 9 stuff, grade 8 and grade 9. Now, grade 12 investigations are not going to be sometimes easy. You need to read into the investigation. But now, you cannot get your variables wrong. Because even in the relationship one, they say it's a relationship between this and that. There are relationship between variables because the more fertilizers, even if we're saying here to determine the relationship between fertilizers and plant growth, fertilizers would still be our independent variable because the more fertilizers we increase or the more fertilizers we add, what happens to the plant growth? It increases. That's the relationship. You eat more, what happens to your weight? It's going to increase. So that's the relationship between food intake and the weight. You eat more sugar and more things like I'm doing currently, I'm gaining weight. That's the relationship between the amount of food that I'm eating and the weight. So it's easy to determine that. Very important. Another thing that I spoke about was the information that you are given. You do not skip that information. You do not skip it. In this information, this was obviously, I think there'll probably be a natural selection or a Lamarck question here because it's speaking about animals, right? Now, in this information, you need to understand how the investigation was conducted. And what are some of the factors that they kept the same? So you need to understand the procedure. You'll excuse me, my phone will probably ring any second now so that I can open the gate for my delivery. Okay, and then you need to look at what we call controlled variables. These are factors that an investigator will keep the same. What is it that has been kept the same? If we say all or whatever, guys, it is kept the same. If I say all my students are attending lessons at 7, that's a factor, meaning all of them are attending the lessons at the same time. Right? Sometimes they will make it easy for you and use the word same. They use 50. That investigation from paper 1, it was a giveaway. It was a giveaway. So if they say all participants did this, they're doing the same thing. If they use 15 male learners, what have they done? They've used the same gender. And if they say all of them are maybe 15 years old, they've used learners of the same age. That is a variable that they've controlled. You highlight anything that you feel like is the same because this is going to be related to a question that they will ask about validity. They can ask about factors that were controlled or what the investigator did to ensure validity. You go back, you look at what was kept the same. So if they say that, yes.
Ma'am, but remember you said we can take our highlighters with us. They don't allow the highlighters.
They don't.
Yes, they don't.
Pencils?
Yes, they do allow the pencil.
Do they allow to write on the question paper?
Yes, that one we can.
Oh, okay. Then use a pencil then. I know at school they allow learners with highlighters. When I was writing, nothing was allowed. You couldn't even write anything on the question paper. But yeah, then your pencil then. Thank you for that. Maybe there's an updated rule. Okay, so use your pencil then or your pen. I'm not sure if they allow you to bring it, but okay, pencil or pen. Okay. You see something like this. There's a point there. These samples were all collected in the morning during summer. That's a validity point. All the samples there, they were collected at the same time or at the same season. I guess since they're saying summer, that's not going to be time. That's going to be a season. So that is a validity point. That is a control point. Or normally they say that is a factor that is kept constant. Okay? You need to get that. Actually, there is the question. There are 2 factors that were kept constant during the investigation. You would need to read. So I've mentioned one. So I haven't read the whole investigation. Then, also this whole thing will also give you information about what we call reliability. And I've said it in the 2 lessons. When you are asked about reliability, think of R as a Republic of South Africa. R, you are looking at whether the investigation was repeated or not. And you mentioned something that is mentioned in the investigation. In the paper 1 investigation, they said it was repeated. I'm not going to go back to it now, but they said it was repeated. So when they ask about reliability, how did they phrase it here? Let's see. I'm a magician. State 2 ways in which the reliability of this investigation was ensured. When they ask about reliability, R, you need to read. Did they say anything about repetition? If they did, that is your answer. The investigation was repeated. If they've mentioned how many times it was repeated, you mention it. So if they said here, I haven't read the whole thing. If they said here, they repeated the investigation 3 times, that is your answer. The investigation was repeated 3 times. Don't just say repetition. What is that? If they did not mention anything about repetition, you cannot answer about that R. The S is about a sample size. A sample size, we are looking at using more organisms or participants or whatever, or samples. So if you collected 1,000 samples, that means that is a large sample size. You used more samples in your investigation. So if they've mentioned that to say maybe 1,000 samples or the insects, let's say they've used 600 insects, that is a large sample size. So what do you say in your answer? They used a large sample size of 600 insects. If they've used 2 boys in the investigation, you cannot answer on the sample size. They did not use a large sample size. They used 2 boys in the investigation. That's not a large sample size. So you cannot answer about S. A is for average. Did they calculate an average? They will tell you if they did. If they calculated an average, that is your answer. They calculated an average. If they did not calculate an average, that can't be your answer. So when it comes to reliability, it's about RSA. When it comes to kept constant, they can phrase it as kept constant, or they can say factors that were controlled, or they can say state 2 factors in which the validity of the investigation was ensured. So kept constant, validity, controlled, it's all the same thing. Another thing that they will ask is your conclusion. Your conclusion will be based on, on the aim and the results. Now, this is obviously a Lamarck evolution, Lamarck natural selection question here. So there's no data, right? So when you are given an investigation, go back to your aim, look at the diagrams that you are given as a result, right? Or look at the table that you are given. So if I were to scroll up, guys, 2.5 hours, my teeth. If I were to do this investigation and say, to determine the effect of fertilizers on plant growth, let's say at the end, I say I've added a fertilizer on my 2, let's say I had 2 plants. I had plant A and plant B, right? And for the same duration of time, I exposed these plants to the same environmental factors, same amount of light. Those would be my control variables, right? If I was pouring water on these plants, I poured the same amount of water, right? Plant A is the plant where I added a fertilizer. So every second day, I was adding a fertilizer on plant A, and I was not adding a fertilizer on plant B. Then at the end of the investigation, let's say I've done this investigation for 10 days. Then I measured the height of the plants. Plant A was measuring to be 10 centimeters, and plant B was measuring to be 6 centimeters, 6 centimeters here. Then they want you to conclude. What have we done? Plant A, we were adding a fertilizer. And at the end of the investigation, the height of the plant was 10 centimeters. Plant B, we were not adding a fertilizer. And what was the height of the plant at the end? 6 centimeters. What was the aim? To determine the effect of fertilizers on plant growth. What will be your conclusion? What can we conclude on here? We can see the fertilizers have had an effect because the plant that received fertilizers has managed to grow taller than the plant that did not. So fertilizers causes plant growth. That is your conclusion. You do not discuss anything on the conclusion. Our aim was to determine the effect of fertilizers on plant growth. Conclude, fertilizers cause a plant to grow, or fertilizers increases plant growth. Those two variables, independent and dependent, are mentioned. Do not say the fertilizers caused an effect on plant growth. What effect did they cause? The plants to grow taller. You can say plants to grow taller, increased plant growth, or any other thing, because even plant B managed to increase in plant growth, but these ones were able to grow taller because they had the fertilizers. Do not discuss anything in your conclusion. Look at the aim and look at the results. This was the aim, effect of the fertilizers. And what are the results? The plant that received fertilizers was able to grow taller than the plant that did not. But that is not your conclusion. If your conclusion does not include both your variables, just know it's wrong. So if you're going to conclude and say plant A grew taller than plant B, that's not a conclusion. You are telling us about the results. Or plant B did not grow taller than plant A. That is not your conclusion. You conclude by answering basically what has happened in the investigation in relation to the aim. Okay. There's other things that I have not mentioned here. Oh, planning steps. I spoke about planning steps. And I said, when you are asked about planning steps, I need someone to remind me. Let me see if you guys were listening. What's the keyword for planning steps? Please tell me. When you answer, decide. Decide, decide, decide. So if I was doing this investigation, I need you guys to tell me. I promise I'm going to be quiet. We're going to end this lesson. But remember, I'm the one who's more tired because I've been speaking for 2.5 hours. But I really am doing this from the bottom of my heart because I really want you guys to do well. If this was our investigation, guys, please tell me the planning steps that we would need to have. This is the investigation. We're trying to determine the effect of fertilizers on plant growth. We've used 2 plants. Plant A is the one that received the fertilizer. Plant B did not. Our duration of the investigation was 10 days. Please give me your planning steps. Anyone? I'll tell you if your planning step is correct or not. Let's speak, guys. There's 13 of you here. Let's speak. Give me one, just one. Okay. Can 5 people just give me only one planning step?
Ma'am, you decide on the method of collecting your results.
100%. Decide on how you're going to collect your results. 100%. Someone else? Even if you haven't said anything the whole lesson, guys, this one is easy. Please just give me a second. Let me answer the security. Hello? Thank you so much. I'll open now. Okay, anyone else? We've said here, decide on the method of collecting the results. Yes?
Could you say decide if the investigation is going to be repeated or not?
That's not a planning step. The minute you say if. Let's try another one. Because remember, guys, I said you use the information that you are given. What would you decide on here?
Ma'am, another one, I think, decide on the type of fertilizer that you would use in the investigation.
100%. 100%. Remember, you are pouring something on a plant. You need to decide. There's so many fertilizers. Which one are you going to use? You decide on the type of fertilizer. What about the amount of fertilizer that you want to pour every day? Don't you think you have to decide on that? You have to. You have to. What about the 10 days?
They also need to decide the times.
The 100%, the times that you're going to pour the fertilizer on each plant. 100%. What about the 10 days? What are we deciding there for 10 days? Decide on the duration of the experiment. So you've decided that your experiment is going to be over 10 days. What about the plants? We used 2 plants here.
Yeah.
We need to
plant.
decide
We
on the
also
type of plants.
yes.
Yes.
Yes. Yes, my people. Yes, yes, yes. Yes. I hope I'll have time to do a calculation tomorrow because they'll probably knock for my delivery now. Guys, I need you to do well. I promise you that's what I want from the bottom of my heart. If it means we need to do another 2 hours, 30 minutes, normally Sundays I prepare for work, but I really want you to do well. That's the only thing that's gonna make me happy, nothing else. Just do your best. And I think that's enough. If you do your best, if your best is 40%, then that's enough. If your best is 70%, then that's enough on my side. So we'll leave it here for today. Please answer the rest of the questions and then let's meet tomorrow. I will say 5 o'clock for now. But if I end up probably being busy about something or not back home, I normally just go to the shops and come back. That's it. So I will see you guys tomorrow and all the best for Monday. We need to push. We need to push. We need to push, my people. We will sleep next week. We're not going to sleep now. All right. Thank you so much for sticking around for 2.5 hours. That's a lot. Bye.
Bye, ma'am.
Bye.
Bye, ma'am.
Bye.
Thank you.
Bye, guys, and thank you.